Hi guys, happy Friday. Can you guys do me a favor and do the last name, first name thing? And cameras on. I just have to set some stuff up with my monitors here. I'll be with you in a minute and then I got to take attendance. All right, looking for Claire to get in. Hopefully her internet works. I am missing Amra, Olivia, Ryan. I know I had an appointment. He emailed me and that's it. Everybody else is here. All right, let me take care of attendance and we'll get going. Amra, Olivia. Oh, we got an exciting lesson plan today. I hope you're as excited as I am. It's gonna be amazing. All right, I'm clicking admit on Claire, but doesn't seem to be working. Send positive internet vibes to Claire so her Wi-Fi works and she can get in. She's trying. Okay, let's go to Google Classroom. Oh, that's screen two now? How's that screen two all of a sudden? Oh, this is screen one? Oh my goodness. Why did I switch everything? All right, let's take a look here. Are you able to see my Google Classroom, or do you just see like a bunch of blue circles and my, my participants list? Which screen do you see?
Google Classroom.
You see the one? Okay, yeah. For some reason, my computer reversed monitor 1 and 2. That was super confusing to me. Okay, so we're going to do a brief discussion of protein stuff still. I told you before that I am expecting you to learn that by watching those videos, which you were supposed to do last week. Hopefully everybody's caught up on that. I know that the beta pleated sheet is kind of difficult, so I want to go over that real quickly with people, and I'll give you a chance to practice that. Okay, Amra's having trouble getting in also. We're going to practice that, and hopefully that won't take too long. We'll see how it goes. I'm going to show you some new tools on the internet that hopefully we can use to help us, because we can't build models in class because you guys aren't here. So we're going to do like virtual models. It's not as good as building the models in class with our hands. We have to assemble everything, but it's sort of a cheap substitute, and hopefully it'll be good. Hopefully it'll satisfy your need for building models. Okay, what I'd like to do first is review the beta sheet, because that's something kind of we went over pretty fast, and the video does go through it and it does explain it, but I want to make sure people are real clear on that. So that's what we're going to do now. Yay. Some of this junk here. I have noticed that when I go into the present thing, my pen slows down and there's a lot of lag. So I don't know if that's a PowerPoint thing or what it is, but well, let me give it a try, and then let's try it and let's see what happens. Maybe it's not going to be an issue, but if it's a problem, then I might go to the other view, which makes everything a little bit smaller for you guys, and we do lose the closed captioning, but we'll try if this works. All right, let's go with purple. Why not? All right, so when we draw amino acids, we have to remind ourselves what the components of an amino acid are. And let me try the letter N one more time. All right, so obviously we have that central carbon. Okay, oops, you lost the pointer. Maybe it doesn't want me to do purple. All right, so I draw the central carbon. I start with that just to give myself a frame of reference to orient myself. A lot of times people draw the R group down, and that's fine, but it could go up also. Like these could be flipped, kind of like my hand could be this way or it could be this way. It's still my left hand. It doesn't really matter. So for this one, I'm going to draw the R group going up. And it doesn't matter which way you start. You could draw it the other way too. It also doesn't matter if you put the nitrogen on the left or on the right, because check it out, my hand could be this way, or my hand could be, okay, that's hard, but it could be this way or this way. It's still my hand. It could be oriented either way, or the pen could be in this direction or in this direction. Same thing with the amino acid. So there is no rule that says the nitrogen has to be on the left. There really is no left and right. So I'll just put it on the left. Why not? Because it doesn't matter. And then there's another carbon here. If I draw the R group up, once these link together in a beta pleated sheet, then the other groups have to go the opposite way. So this has to go down. Obviously, this one goes down. And then the double bond O goes down. So if the R group goes up, all the rest of the stuff goes down. If the R group goes down, all the rest of the stuff goes up. So then I'm going to link the next one onto it. And that has to be now we're fixed. Now we have to have the nitrogen here first, then the next carbon, then the next carbon. And this time they're going to flip. So when a beta pleated sheet forms or when an alpha helix forms in secondary structure, these will flip around. So this will now go down. That means all of these guys have to go up. They have to go up and up. And I'd like you to practice this a little bit if you haven't already. I'd like you to practice drawing this by memory. Because eventually we're going to have a test. And I'm kind of thinking about, maybe in about a week and a half or so, I still want to talk about enzymes. I want to do a virtual lab with you guys. And we still have to do lipids and DNA, but that won't take too long. And then after enzymes, we'll have a test. So a week and a half-ish. I don't know what date that puts us, like 15th or something, whatever the date might be. On that test, I think what I'm going to do is have you guys I really haven't figured this out yet, but what I'm thinking about is I'm going to have you guys put your Chromebook on your desk, like a little bit further away, so I can see you drawing. I can see you, drawing on paper with pencil. Then you upload that with your phone and turn that in the Google form. That's how we did tests and stuff last year, but everybody was sort of on their own doing it, without me supervising. This time, well, I'm hoping to make it closed notes. I know people could still cheat, but you guys are honest. I can trust you guys. And if I tell you ahead of time, you got to have it memorized. It's not that hard to memorize. You can just do it. It'd be easier to memorize it than to set up some elaborate cheating, paradigm that would actually take you more time. It'd be more labor intensive. So Noah's doubting me. You could beat my system, Noah. Yeah, I'm sure you could. But anyway, do you want to use your powers for good or evil? That's a decision you have to make. So you guys decide what you're going to do. I'm hoping you choose good. All right, next one. Flip to. Did I lose my pen? No, still have it. All right, what's going on now? Amra's here. Yay. Okay, Amra, and then we're trying to clear one more time. Okay. Anyway, we got N, we got C. Ooh. And we got C, and this R goes up because the other one went down. This one goes up, and therefore our hydrogen is going to go down. This guy is going to go down, and then the double bond O is going to go down. And that's just going to continue like that in this direction, and this might continue in the other direction. So as they curve around and they're underneath here, now when the nitrogen was on the left up here, now the nitrogen has to be down below, and it's going to be in this orientation. If this double bond O is pointing down, then this hydrogen has to point up on the amine. Well, it's not really an amine anymore, but on the nitrogen. And then this hydrogen is going up, and this R group's going down. So I'm just reversing everything. I'm going kind of backwards. And notice how I don't have to memorize all of this. All I have to know is one amino acid. I have to know that it's carbon, carbon, nitrogen, and that there's a double bond O and a hydrogen. If I remember that, all I have to do is flip these around. And it's actually pretty easy. And I should line this up. So this nitrogen has to be just below this carbon. This nitrogen has to be just below this carbon. N, C, C, going backwards. Now the R groups are both pointing in. This hydrogen is down, this double bond O, and so on and so on. It just keeps going until we're done with the protein. And we're not going to draw the whole thing because that would be crazy because proteins are really big. But we're going to draw like 6 of them here. So that'll be nice to show how these link together. And because it's secondary structure, we said in secondary structure, it's just hydrogen bonds. By the way, the pen works fine in present view. I don't know what I was talking about before. When I was trying it earlier today, it was going crazy. It wasn't working well. Secondary structure is hydrogen bonds betwixt atoms of the backbone. Now, that's not very specific, atoms of the backbone, but what we're talking about, not the R groups, atoms of the backbone. So the double bond O and the hydrogen. The hydrogen of the nitrogen, though, not this one, and this one, this one, and this one. And those are going to form hydrogen bonds between them because this is slightly positive and this is slightly negative because oxygen is electronegative and nitrogen is electronegatives, making this slightly negative here, but that hydrogen is slightly positive. And this carbon is slightly positive here. But that doesn't matter so much. It's this positive here and the negative here that matters. So we'll get a hydrogen bond here, here. I'm drawing a dotted line because it's a hydrogen bond, not a covalent bond. Yeah, we are honest, Mr. Volk, don't you worry. I know you're honest, Sim. You're like the most honest person I've ever met. The most trustworthy. It's that face. You can just tell you're a good person by the face. Anyway, am I allowed to drink Diet Coke during class? Okay, I'll just do it off screen. Here we go. That was not Diet Coke, don't worry. That sounds so creepy. My Diet Coke thing or your honest face? Anyway, whatever. So that's that. All right, let me page through the rest of the PowerPoint. I don't know if there's anything else. So quaternary structure is just, you know, if you got your tertiary structures that form a lot of times it's 4. Quaternary, by the way, does not mean there's 4 subunits, just means it's the 4th level of structure. It could be just 2, or it could be 3, or whatever. And these will link together, you know, kind of like hemoglobin. All right, cool. And we practiced this with that group activity we did last week. So, but I'd like you to review that and study that and kind of know that by heart, primary through quaternary structure. Including all the linkages of tertiary structure. You know, you got your disulfide bridges, you got your ionic bonds, and which amino acids do that, you know. Ionic bonds, it's the plus and the minus ones, the acidic and basic. Disulfide bridges, it's cysteine. You got your hydrogen bonds between polar ones. You got your hydrophobic interactions. You got your van der Waals forces. That could be between any of them. We don't point any particular ones out, but the hydrophobic ones, obviously, it's the nonpolar R groups. So you got to kind of know that by heart. Oh, we can just cheat and look it up when you give us a test. Yeah, well, we'll see. We shall see. You have to live with the guilt, boys and girls. Anyway. What am I doing? Okay. That just gives you kind of a fancy picture of all the different linkages. Although, do they? No, they have all 5 here, yeah. Oh, wait, do they? Hydrogen bond, hydrophobic, disulfide, ionic. Oh, they don't show us van der Waals, so that is missing. Okay. Just examples of collagen, hemoglobin. We talked about those in our group thing. Here's some hemoglobin stuff. Normal hemoglobin, beautiful. Look at that globular structure, alpha and beta. Perfect. Carries oxygen. We love it. And then like goofy, horrible, twisted hemoglobin in sickle cell that causes it to link together and form these long rods that are not supposed to form, which then makes the cells shaped like a moon or a sickle. My goodness, that's how they look under the microscope. Looks scary. All right, proteins denature if they get heated or if there's salt or if there's acids. Basically what happens is, this I actually maybe should elaborate on a little bit. If you have linkages in a protein, so let's say here we have ionic bond right here, or here we have some, hydrogen bonding going on. If that's the case, if you add acid, if you put an acid in there and acid has a whole bunch of hydrogen ions, those hydrogen ions are going to go in and they're going to stick to the negative charge. Those are going to come in there and they're going to stick to this negative charge and they're going to block that from then bonding to this, which is going to cause it to come apart. Same thing with salt. So if you put a strong salt solution in there, sodium ions and chloride ions or any negative ion is going to get in there and mess around with the positives, and this positive ion is going to mess with the negatives and it's going to break these up. It's going to get in between there and destroy that. And that's why low pH or high pH denatures proteins and destroys them because they get in there. Heat destroys proteins because basically heat is just molecular motion. So when the molecules are moving faster back and forth, and water molecules are coming in and bombarding this thing at high speed because it's hot, and the water molecules are moving faster, they slam into this like little bullets, and they destroy the protein. They break it apart into this denatured structure, which is ruined, essentially. It's broken. All right, so salt solution, pH, fluctuations, heat, all causes denatured proteins. And we know this, like when we fry an egg, the heat does that. Okay. Chaperones I talked about in the video. Let's talk about lipids. Did I already? We didn't start lipids already with me lecturing, right? I've not talked to you about lipids at all yet. Am I right? Okay, well, we're gonna do lipids. So lipids are basically fats and oils. And we know what those are. Fats we know are solid at room temperature, and oils at room temperature are liquid, like corn oil or olive oil or something. And fats like, you know, Crisco or butter, or like the fat that's on meat, like on bacon or something like that, solid at room temperature. But other than that, they're very similar in structure. There's some slight differences that causes them to be either solid or liquid, which we'll talk about. Just looking at the chat, it's just foolishness in the chat, I guess. Is Claire in? Did Claire get here? No, she's not in. Oh my goodness. Okay, well, I'll post. All of these are, I have videos of all of this stuff that I'm posting. And why don't I just show you something for just a second, since I just remembered. Sorry to be so disjointed here. But let me just show you something on my what I did on Google Classroom today. I just did this today. I just created this today, and I think this will be useful to those of you that want to review or work ahead, or sometimes I'll assign videos when I actually won't talk about it live in class. I might just assign some. Like, for example, in like 2 weeks, I'm going to have a medical thing where I won't be here on a Tuesday. And I'm going to assign you videos to watch that you have to do on your own and take notes like I did last time. And then you guys didn't do it. This time you're actually going to do it. Okay. Last time that was my fault. I didn't say you should really, really do it. I just said do it. And that wasn't like clear. But this time I'm saying really, really do it. And then you're going to do it. It's kind of like when my wife talks to me and she's like talking and I'm not listening. She's like, I need you to listen. I was like, oh, okay. Now I'm actually listening. So see how good of a husband I am? How I pay attention? All right. Anyway, I'd made this. Check it out. And it's not very attractive or anything, but it kind of summarizes all the videos that I've created that are on EduVision. So chapter 3 ones over here, 6 of them. There's only one chapter 4 one. It's kind of long, but there's not that much in chapter 4. So it's like 20 minutes or something. And then at chapter 5 is the big one. That's lots of material, carbohydrates and proteins and lipids we're going to talk about today. All the videos are organized here in sequence. And as I create more, I'm going to keep adding more. Does that make sense? Because so far what I've been doing is just posting on Google Classroom like, oh, here's some videos, and then here's some more videos somewhere else, and then here's some more. It's kind of hard to keep track. Where are these all? So here you have them all in one place. You appreciate this, or is this like not something you care about, or is this good, or feedback? Give me a little. I'm just a very needy person. I just need to be praised sometimes. So, you know, I got self-esteem issues. So anyway, wow, I got some really enthusiastic thumbs ups. Thumbs ups. What's the plural of that? thumb ups, whatever. Anyway, okay, back to lipids. Screen 1 now. So confusing. Oh boy. Alrighty then. Well, okay. Inevitably, almost all lipids in living organisms, whether it's in your body or whether it's in food, are triglycerides. Just about all lipids are triglycerides. And actually, they don't say triglycerides here. They say triacylglycerol, which is the same thing, just a different name for the same thing. Hold on, I'm losing control of my mouse here. And my pen's attacking me. All right, let's try that again. So this triacylglycerol is actually pretty good. It's a pretty good description. The tri means 3, the acyl means acid, and the glycerol means glycerol. All right, so I call them triglycerides, which is also acceptable. Either way, both of these terms mean the same thing. So what are triglycerides? Well, they have a glycerol, which looks like this. Disturbs me when I hear loud noises in my classroom and I'm the only one in here. Okay, so the glycerol is 3 carbons. It's classified as an alcohol because it has these hydroxyls over here. Alcohols are hydroxyls stuck to a carbon. So this is a type of alcohol. It's not the alcohol that's like in wine and stuff like that. All the rest are hydrogens because all the carbons have to be kept happy. They have to have 4. So that's what glycerol looks like. All right, and then that glycerol is going to link up with 3 fatty acids, and those are going to look like this. They're going to have a carboxyl on one end like this, and then a carbon chain. And I'm going to be lazy, Volk, and I'm going to only draw a little line for the hydrogens, because otherwise this takes really long. And so then I have 3 of these that are going to link up here. And how long you make these, that's up to you, because so the longer you make them, the more likely it's going to be a fat. The shorter you make them, the more likely it's going to be an oil. If they're really, really long chains, they tend to be solid. They tend to be fats. And if they're small, they tend to be oils, liquids. Okay, and we'll do one more. And which direction you put the double bond O doesn't matter, because those can spin, because it's just a single bond on that carbon chain. And, whoops, forgot one here, like that. All right, so we'll put those hydrogens in, just being kind of lazy here. Sorry that got messy, but whatever. So then these need to be linked together. So I'm going to leave that to you now to link these together, but in order to link them together, we have to do dehydration synthesis or condensation reaction, which means water has to come out. And do you see water maybe 3 times here? Would you be able to find water 3 times? I'm just going to change something here on my screen. No longer see you guys. I don't know why. All right, can you see water? Yes, no? Everybody say yes.
Yes.
Give you reassurance you are appreciated. Oh my goodness, I got a little tear in my eye. All right, so I am seeing maybe a water here, here, and here. And then what we can do is we can get rid of those waters and then link them together. Okay. So anyway, that's what's going to happen to form that triglyceride. So we got 3 fatty acids, 1 glycerol makes the triglyceride, and then it ends up looking like this. So you have an ester linkage here. This is your ester linkage, where there's a double bond O on a carbon and then an oxygen. And obviously they're all the same here. And then there's your fatty acid chain from your fatty acids, your carbon chain from your fatty acids. And here's what used to be the glycerol. I mean, you can't really call it glycerol anymore because it's now like, it's not OH anymore, but anyway, we get the idea. And here the water comes out, dehydration synthesis, do that 3 times and you form that. Okay, so sometimes if I want to be super lazy, I'll draw a triglyceride like this. It implies that this would be the glycerol and these would be the 3 fatty acids. Okay,
I have a question.
that would be a grotesque abbreviation if I did that. Go ahead.
So like, you know how you said like how if like the fatty acid chain is longer, it's most likely to be like a fat. So like if you were to ask us to draw a fat versus an oil, like how many like carbons would you put like after like the HO, like for the fatty acid chain?
That we're not going to worry about, but it ends up being like somewhere between maybe 8 and 20 for the,
Okay.
sorry, that's for the fat, but for the oils, they're shorter, like maybe 4 to 6 to 7, 8, maybe something like that. And there's a cutoff where it turns to oil, like where it turns to liquid and where it turns to solid. There's like, that's known, but I just don't have that memorized. So if I don't know it, then you don't have to know it either, which
Okay.
is kind of a cheap statement, but it is what it is. You know, if you had a better teacher, then you'd have to learn all that stuff, because if the teacher actually knew anything, you'd have to learn way more. So lucky you, you have me, right?
Yeah.
Yeah. All right. Okay. You know something? We should probably learn line diagrams. I never taught you line diagrams. Let's see if I got any space here. Oh, there's some well, it's quasi line diagrams. So here's just another example of a triglyceride, saturated fat stearic acid, which makes butter. Stearic acid is the fatty acid that's on this triglyceride, but all right, this goes into cis and trans, which I don't quite want to talk about yet. Let me do something here. Let's keep these for now. We're going to insert a blank page so we can do some doodling. All right, let's see what we can do here. So when I draw a triglyceride, here's what I would, oopsie doopsie, pen, interruptions. PowerPoint is not super virtually friendly with tablets, but it's good enough. All right, normally what I would do is draw this, and then I would put that ester bond in there, and then I would do my carbon chain. And that, you know, that takes a long time to draw this. Like if you're going to draw it all out, oh my goodness, it's a pain. So we need a shortcut here to do this faster. We do need a shortcut. Okay, no one's in the waiting room. And you know, obviously I'd have to draw the hydrogens in. Oh my God, what a pain. Okay, so one thing we can do is we can draw carbon chains like this, first of all, okay? We can draw them like zigzag lines. Each corner, like this would be a carbon, this would be a carbon, this would be a carbon. And the hydrogens don't even get drawn in at all because they're implied. Because people that know biochemistry and organic chemistry, they know that there's hydrogens here. They know there's 1, 2 here. They know there's 3 here, et cetera, et cetera. So that's a much faster way to do it. And you don't even draw the like circles in here. You just literally do this for the carbon chain. So this would be like 1, 2, 3, 4, 5, 6, 7. This would be heptane right here. Okay, so how do we do this now, which is a little more complicated? Well, let's check it out. Let me just see if I can erase this real quick here. Oh yeah, that's nice. So nice. Let's go back to our color. All right, so how am I gonna draw this? Well, I'm gonna do this. So that's my glycerol. I'm like, what? How's that glycerol? It's just kinky line. Well, here's a carbon, here's another carbon, here's another carbon. So it's 3 carbons there, 2 on the end and 1 in the middle. Okay, then I'm going to draw my oxygen in there like this, and then I'm going to draw my carboxyl like this, and then my carbon chain. So how many did I have here? 1, 2, 3, 4. 1, 2, 3, 4, 5, 6. I put too many, so I'm just going to add these here. So that'd be that top one. Okay, so then here's another, oops, here's another oxygen, and then I would go like this. 1, 2, 3, 4, that's this one. And then I would draw another oxygen, that's this here. And then I would do this. Oops, I don't know what it always does that. 1, 2, 5. So 1, 2, 3, 4, 5, double bond O. That's how I would do that. So this is a line diagram, super fast drawing of a triglyceride. Now it took me a while because I was talking, but if I just had to draw that without interruption, like if I don't have to like explain it to people, I would be able to draw that much faster. So it's kind of a nice system that we can use to draw these molecules more quickly. Like a fatty acid, I would just do this. Just for a fatty acid, just do that. Just like that. Super fast versus drawing all the carbons and hydrogens in. You know what I mean? I hope you understand that. I hope you appreciate that. So we call that line diagram. It's just an abbreviation, and it assumes hydrogens are in there. We can put double bonds in too, like this, you know, put one here. Then now the hydrogen number changes because there's a double bond. This carbon here and this carbon here already has 1, 2, 3 bonds on them. So this only gets 1 hydrogen. This only gets 1 hydrogen. This one has 1, 2, and then that would get 2 hydrogens then, like that. This one only has the 1 covalent bond here. That would get 3 hydrogens. So that's something I'd like you to be real good at, is understanding the hydrogens and the valence of carbon and all of that stuff. So anyway, okay. I think it's time for us to talk about trans and cis. I think that'd be a good idea. So one thing that determines whether lipids are solid or liquid, whether they're fats or oils, is the length of the chain. Okay, so long, fat, short, oil. That's just a trend. And once again, the actual number, who cares? I'm not going to worry about that. We're not going to memorize like specific fatty acids and their length, like how many carbons they have. We're not going to do that. Why? Because I don't know them. I don't have them memorized. I know some, but I don't want to memorize all those, so I'm not going to make you do it. Okay, so that's saturated. If they have double bonds, then it's unsaturated. So notice this one has these guys have all single bonds here. There's no double bonds anywhere. It's none of this here. Okay, none of this going on. Undo that. For some reason, PowerPoint doesn't let me do Control Z to undo. That'd be nice. I have to actually go to the eraser. It's kind of funny, but whatever. All right, anyway, let's go green here. All right, sometimes there are double bonds, so then we say they're unsaturated. So if we throw a double bond in, it's unsaturated. And a lot of times, unsaturated tends to be more on the liquid end. It tends to be more driving it to be a liquid. So the ones that are very unsaturated tend to be more liquids. The ones that tend to be saturated tend to be more solids. So there's 2 things that make a lipid a solid or a fat. 1, long chain, and 2, saturated. Okay. Anyway, a lot of times when they're unsaturated, they get a kink like this. Notice how it goes this way and then it kinks like this. That's a cis double bond. If the double bond causes a kink, it's cis. And the reason why that is because the hydrogens on this double bond are both on top here, and it causes it to, now this should be a more extreme kink. It doesn't show the kink very well here, but if the hydrogens are both on top, they kind of fight with each other and they're in each other's way. There's competition for space here. So then they drive each other apart a little bit and pushes this down. So that causes the kink. We call that a cis double bond. Remember we talked about cis and trans before when we talked about stereoisomers? Yeah, we talked about hydrogens being top and bottom and all that. A trans double bond, that's this one, would be on top and then on the bottom. So the hydrogen is one is up and then one is down. And that's a trans configuration. And the reason that's called trans is because the word trans means opposite. Or it means does it mean opposite or across? Maybe it means across. I don't know. One or the other. Anyway, so that's the trans then. So that's another, so both of these are unsaturated. Both of them are unsaturated. This one has a kink. This one doesn't. Cis has a kink. Trans doesn't. This one tends to be healthy to eat. These are healthy lipids, healthy fats to eat. Like, you know, stuff in like olive oil or canola oil or fish oil. These are good ones, avocados, nuts, things like that. These are unhealthy. Here they showed a little kink, but it's basically mostly straight. It causes the chain to be straight. And if the chains are straight, they tend to pack together really tightly, which makes them more solid, if they can pack together. But if the chains are kinked and they're all over the place like this, well, kinky like that, and they don't fit into each other very well, then there's going to be bigger spaces between them, and that's going to make them more likely to be a liquid. The kinkiness doesn't allow them to pack so tightly like these guys, making them more likely to be a liquid. That makes sense? If I lean back like this, can you still hear me okay? I'm far away from the mic.
Yeah.
Yeah.
If you see me like get too relaxed by leaning back and then falling asleep all of a sudden, then you got to yell and wake me up, okay? Hello?
I guess. I mean,
Yes.
could wait like 10 minutes and then wake you up.
Thank you. All righty. Oh, more of the same, cis and trans. Why do I have 10,000 pictures of the same thing? I have no idea. Kinky, not kinky. Oh, trans fats are in like, okay, so the unhealthy ones tend to be in like, they're not really that natural. Trans fats don't exist very much in nature. They're usually created by people, by food companies for like processed like stuff like, I don't know, store-bought cookies or Hostess snacks like the Twinkies and all that junk and Little Debbie snacks and all that. So store-bought cookies and things like that have a lot of trans fats usually, and that's unhealthy. We used to think that anything unsaturated was healthy, but it turns out the trans unsaturated lipids are really unhealthy. They're bad. They're like almost worse than cholesterol and the animal fats. Animal fats aren't trans. They're not unsaturated. Animal fats are saturated. They all have single bonds. Not all, but most animal fats, like chicken fat, pork fat, beef fat, stuff like that, is saturated, doesn't have any double bonds at all. This stuff is made out of plant fats that have been sort of artificially changed to be put into these cookies and like Chips Ahoy and Keebler and Oreos and stuff like that to make the cookies taste better and to make them last longer on the shelf. It gives them a better shelf life. So while these are delicious, they're unhealthy, sadly. All right, how are we doing? Oh, what time it is?
Good.
What time is it? Well, we got till 1, so I want to have you guys do a little model building here, but before we do, I got to teach you about phospholipids. Then we'll do, then we'll mess with beta sheets, which maybe I should have done before we started lipids, but oh well, this will be more confusing, which is good for you. And then we'll build a cell membrane, which will be nice and easy. The beta sheet will be a lot tougher. All right, so I said the cell membrane is going to be nice and easy, and then I throw this picture up here, which looks horrible. It even has a scary monster here that looks like it's going to, like a space alien that's going to attack us here, but this guy. Anyway, so this is basically the, it's sort of a hybrid line diagram and then, drawn out structure. And this is the space filling, like what the atoms would look like in 3 dimensions diagram. You don't really need to worry about this, but notice let's start with the glycerol. So the glycerol, what used to be glycerol is right here. That's right here on this guy. That's this. That's what used to be the glycerol, those 3 carbons. Okay, so phospholipids are basically triglycerides, just like what we just learned with the glycerol and everything, except they only have 2 fatty acids coming off. And then instead of a 3rd fatty acid here, that's chopped off, they get a phosphate. All right, so they get a phosphate. And then this thing up here is like an extra thing stuck to the phosphate. We don't really need to worry about that. Here, there's a choline. That's pretty common that they have like a hat on top of the phosphate. In membranes, that you see that quite a bit. Phosphatidylcholine. Oh, sorry, I shouldn't throw words like this. One is called phosphatidylcholine, but this one is pretty common in cell membranes. It's kind of one of the famous ones. I'm not going to worry about this right now. I'm just going to worry about the phosphate, the glycerol, and then the fatty acids. So let's try drawing that. Oh, I thought I gave myself I got to make another slide. Sorry, I thought I did this already. I guess I didn't. Ah, boy. No slide. Yada, yada. Go back to the presentation. I have a different version of this PowerPoint that actually does have it, but oh well. Let's go blue. So let's draw the phospholipid. We're just going to draw the glycerol this way. So I'd like you to also draw this so you learn this. Because on your closed note test, you're going to have to know this, yeah. And we're going to pop these hydrogens on here. Then we're going to put the fatty acids down below. La, la, la. So we're flipping it sort of sideways now. And I'm just going to do this lazy line diagram thing here. And we got the ester bond here, yada, yada. And then we'll do a lazy guy like that. So that's our carbon chain. This is our fatty acid 2. We got 2 of them. And we don't have a third one. That's just a hydrogen now. And instead we have a phosphate here. So we're going to put an oxygen from the phosphate and then a P and then another oxygen and then 2 more. So there's 4 total. This has a minus, this has a minus, this has a double bond. No minus here. Because oxygen has to have 2 bonds. There's 1 here, and then the negative is where the other bond would have been from the hydrogen. Same thing here. And phosphorus likes 5, actually. So we're not going to worry about the valence of phosphorus. That gets into some inorganic chemistry we're not going to concern ourselves with. We'll let Carruthers and those other chem teachers worry about that. Fangman and them. What do you guys have for honors chem? Who's teaching honors? I don't even know. Shane?
Yes.
Shane?
I have
Sure.
Fangman.
So you like half and half? Some of you guys are one, some of the other. Okay, that's great. All right, they'll teach you, they'll learn you good, all that inorganic chem stuff. Okay, so we have a phosphate, we have the glycerol, and we have the 2 fatty acids. All right, well, I don't want to draw all that every time, so we have an abbreviation. We just draw the phosphate and we draw the 2 fatty acids like this. What about the glycerol? Yeah, that glycerol is in here somewhere, you know, it's in there. So that's how we draw phospholipids if we want to do like the really abbreviated cheap structure. If you draw it out, you have to draw all of this. I guess we could pop these in here. So anyway. All right, so practice that. My video that I posted on this, exactly the same thing as what I just did now, walks you through that, and you can watch that again if you missed this or whatever. All right. Also, I think I'm recording this. Am I recording this? I don't know. It's supposed to be recording.
It says you are.
Oh, why doesn't it tell me that? Okay, anyway, I guess I'm recording. All right, phospholipids. So in a membrane then, these little guys are gonna do this. These little guys are just gonna line up like that with all the fatty acid tails in the middle and all the phosphate heads on the outside. And the reason why is because these phosphate heads are charged. They have negatives on them. And that makes them super hydrophilic. Hydrophilic means loves or hates?
Loves.
Loves water. Loves water. And the fatty acid tails are super hydrophobic because they are nonpolar. They just have carbon and hydrogen in them, nothing else. Carbon and hydrogen is not electronegative. It's going to be nonpolar. It's going to be hydrophobic, and they love to bunch up in the middle. So they stay in the middle, away from the water. They're sheltered and protected, and it forms this spontaneous sort of bilayer. I'm drawing blue on blue. That's real smart. A bilayer, which means 2 layers of these sort of oppositely oriented, like this, phospholipids. Okay. And obviously in a real membrane, you'd have like proteins in here, you know, and like all kinds of like peripheral proteins and then integral. We'll learn about that again. We'll cover that again, but you know this already from freshman bio. The fluid mosaic model. Ooh. All right. So then, yeah, I really messed up with my, my other version of this totally had like blank slides, but then I didn't use that one because I'm dumb. So we're going to insert a slide and go back. Do you enjoy my struggles?
Yes.
My suffering causes you pleasure? All right, so there's another molecule we're going to see in membranes sometimes, and this is like a whole nother class of molecules. So this kind of is a segue between phospholipids, and the next third class of molecules that we're going to discuss, which is the sterols. And the most famous of the sterols is cholesterol. It's the primary one, the most common one that we're going to deal with in our bodies. And cholesterol kind of got a bad name because cholesterol has been associated with disease because people eat too much cholesterol. Oh, Ryan's here, nice. So people associate cholesterol with, heart disease because it does. And if you eat too much, it clogs up your arteries and can cause heart attacks and all this. It is. Hi, Ryan. Welcome. It is a bad thing. I mean, it's not, it's not unjust to malign it like that. But cholesterol also has actual functions in the body, in animals and in humans that are pretty important. So let's draw the structure of cholesterol. I'm going to give you, so here's the real structure, which we're not going to worry about. That's cholesterol. So you're not going to memorize that. We're going to memorize a cheap symbol version of it. So the way we're going to draw it is like this. We're just going to do a hexagon ring, carbon ring, another one hexagon ring. That's easy. Just stick it together. Another one hexagon ring. Oh, 3 of them. My goodness. And then a pentagon ring. And then up here it gets an OH, hydroxyl. And down here it gets this chain. It's some hydrophobic carbon chain, nonpolar carbon chain. So that's cholesterol. Okay, so 2 functions that cholesterol has is 1, steroid precursor. So we use it to make estrogen and testosterone and other steroids in the body. And notice how the structure of estrogen and testosterone is really, really similar to cholesterol. It just has that chain missing, some minor modifications. Also, estrogen and testosterone are very similar to each other. So one of them is like what we call the male sex hormone, and the other one is the female sex hormone. And you're familiar with the word estrogen, and this says estradiol. Estrogen is more of a class of molecules. Estradiol is the predominant one. That's the one that when people say estrogen, they really mean estradiol. That's the main one. Okay, so that's one function. And then another function of the cholesterol is this. It is, let me see if I got a blank slide back. No, I don't. I'm so stupid. Another one in there. You're a very forgiving audience. Or maybe you're cursing at me, but you got your mic off, which I appreciate if you got your mic off for that. That's lovely. All right, so, we draw our cell membrane, we got our phospholipids. They're all buddies like that. They hate that water down here, and they love the water up here. Here's the water. They're doing all this stuff. But then sometimes right in the membrane, you'll see this. Oh, well, what do you know? Who invited this guy? Okay, so he's like just hanging out with the phospholipids. He's like just one of the phospholipids here, and there might be another one down here. I'm just going to do a quick version of it. But the cholesterol integrates itself into the membrane. And what it does is it stabilizes the membrane. So at high temperatures, the membrane tends to get more fluid. The hotter it gets, the membrane becomes more fluid-like, more watery. And it gets too hot, the membrane falls apart. It can break apart because it's too fluid, watery, fluidy, I don't know what the adjective is there, but anyway, too fluid-like. And at low temperatures, the membrane tends to freeze. So if you take if you take some corn oil or something like that and put it in the freezer, it'll actually turn solid. So if you lower the temperature too much on a membrane, it'll turn too hard. It'll stop being a liquid, which is a problem. So at low temperatures, that's a problem too. So what cholesterol does is it stabilizes and keeps it kind of in the middle. At high temperatures, it prevents it from falling apart. It keeps it together. It prevents it from becoming too liquid. At low temperature, it prevents it from becoming too solid. So it stabilizes the membrane no matter what, at low or at high temperatures, which is actually great, which is actually a cool thing. HDL is not actually cholesterol at all. Oh, somebody asked, when you ask these kinds of questions, it would be actually better if you asked them public, because then everybody could see it and we could all learn from it. So somebody asked, she says no, okay. HDL cholesterol, is it bad for you? Yes. So here's the deal. HDL is a high density lipoprotein. It's different fats like fatty acids and triglycerides and stuff stuck to a protein. And HDLs are actually good for us. They're like the, what do you call them? Like the cis guys. LDLs is low density. Those are more like the saturated ones that bind with protein and trans ones. And the LDLs are unhealthy and cause the heart disease. And the HDL actually fight the, okay, that's not true. They don't fight, but they offset. Like the higher your HDL is, the better. And the higher your LDL is, the worse. And the higher your HDL tends to push down the LDL, which is kind of nice. So if you eat fats like fish oil and nuts and things like that, that's good. Obviously, there's limits. You can eat too much, but I mean, you know what I mean? All right. Okay, we don't have a ton of time now, and I talked a lot, but what I want to do now is stop, and I want to guide you to the Google Classroom. I'd like you to look at something. I want you to, this is going to be a little bit confusing at first, but I want you to give it a chance, please. I worked on this morning, and it was kind of stressful, and I hope this works. I want you to use Jamboard. Have you guys ever used Jamboard in a class? Jamboard's pretty fun. It's nice. Have you guys used that? Okay, it's like a whiteboard. You can do a lot of stuff. It's easy to use. It's real simple, and you can like just slide things around and build things or whatever and display stuff. You can work together with each other, which is awesome. Okay, so you're going to open a Jamboard like this, and then I'm going to put you in breakout groups, and one person in the group's going to basically copy this. So you're going to go in and you can make a copy of this. You can do that right now, but only one of you will need it. So if you do one now, you might not use yours. Then you're going to share your copy with everybody else in your group. So what you can do is go to share, You can go to get link. Anyone on the internet can view. So if yours is restricted or whatever, make sure it says anyone on the internet can view or whatever, and then make it editor. I want you to make yours editor so that other people can edit like this. So I'm not going to do that because I don't want you to edit mine, but you're going to let the people in your group edit yours. Does that make sense? Then you're going to copy the link. And you're going to put it in the chat of your group, of your little group. And then everybody can go on that Jamboard and work together at the same time. So then what I want you to do is I want you to, this part is the part that's going to kill you. I want you to go to Sketchpad. I showed you Sketchpad before, right? I hope. If not, just go to Sketchpad, okay? I gave you a link, I think. So I'm going to go to my Sketchpad here. Okay, there it is. And I want you to draw an amino acid with your mouse. By now, everybody has a mouse on their Chromebook. You're not still using the touchpad, are you? If you are, shame on you. Get a freaking mouse, for crying out loud. Or get one of these. This is even better. You know, these, that's what I'm using because I'm pro. So anyway, I want you to draw an amino acid. So what you're going to do is go to Sketchpad, and there's nothing to install. It's just a website where you can draw. It's really nice. No comment. I'm going to go over here. I'm going to go to pen. So I'm going to click on this feather thing. I'm not going to do calligraphy. I'm going to do pen. And then I'm going to draw a friggin' amino acid. I'm just going to go like carbon, carbon, nitrogen. Let's go down. I'm going to do that. And you're going to do that. Now it's going to be harder for you because you're using the touchpad or the mouse. And it's not going to be as easy, but that's what you're going to draw. Put a little like bond here and a bond here, and then you're done. I'm going to get a mouse. Okay, that's fine. Okay, so that's what you're going to do. Then you're going to crop this as tightly as you can. You're going to crop that. Okay, so as tightly as you can. So just do your best here. I'm going to crop it real tight on this end, just like right to the edge. Apply crop. And then I'm going to do it again on the other side. So I'm going to start down here. I'm going to crop it like super. So there's nothing extra, no extra white space. Bam, crop, bam, good. Now I'm going to save it. Actually, you could like do this while you're watching me. If you want to, you could do this. Now I'm going to save it. So on the bottom left, there's like an export thing here. I'm going to click on that. I'm going to save it as PNG or JPEG. Don't do PDF. I don't know what this is. Either JPEG or ping. I'm not going to do ping. All right, cool. Now it puts it in my Google Drive. Actually, here it puts it on my hard drive, but for you, it's going to put it in the Google Drive. On the Chromebooks, it's going to put it in your Google Drive in the main directory, and it's going to be some horrible file with like numbers and letters. Okay. So then you go back to Jamboard, and you're going to go to the next page, and you're going to import that amino acid. So you're going to go to Add Image. You're going to go to Google Drive, and then you're going to pull it up. Boy, that takes long. Why does it take so long? Okay, there it is. So there's okay, that's not actually the same one I just drew because that's another one I made earlier. But yours is probably going to be like that because I drew this one with a mouse. It's kind of ugly. All right, so then you have that. So then you need one more. That's the reverse. Okay, back to Jamboard again. I mean, sorry, Sketchpad again. Now we need another one. So I'll make a new one. New, blank, go to pen, and make another one. But this one I want reversed. So did I have it NCC or what did I have? I think I had NCC. Did I? Yeah, NCC. Is that what I had? Anyway, I can just check here. Well, whatever. Where's my guy now? This guy. All right, so this time I'm going to do it backwards. So I'm going to do N, C, C, and this time I'm going to put the R down here. I'm going to put this up. I'm going to flip it, basically, is what I'm saying. I want it flipped. Okay, and I'm going to put the little bonds in. Yep. And then I'm going to crop it as well. That's pretty tight already, so I'm going to leave it the way it is. I'm going to save it. And now that's going to be on Google Drive, and then I'm going to import that into this. All right, so here we go. Oh, I have to move mine around. Yours will be in Google Drive. Mine isn't because I'm on a computer. So then you would import that. And then you can make some copies of these. You can duplicate them. You can scale them. And you can flip them for the one below. And then you can line them up like this. Look at that. You can make a beta sheet. You just need one of each, the flip one and the normal one. So you make yourself like 4, 6, 8 copies. As a matter of fact, what you're going to do is every person in the group is going to import one, at least one, and then make a copy of it, and then you have 2 of those. So then we're going to have a beta sheet that's going to be like a chimera made from all the different amino acids that you guys drew in the group, which will be super fun. How much time? Oh, we still have 20 minutes. We can do this. We can do this. Yes. Okay. So I don't want to ask if anybody doesn't understand this because I'm going to get 30 people telling me, oh, I don't get it. So I'm just going to like pretend you got it. I'm excited. Look, I mean, this is fun, isn't it? He's so hyped. Yeah. That you could have typed privately. Oh my God. Anyway, not fun. We love the enthusiasm. Yeah. At least one person likes it. That would be me. So I want you to import a bunch of them. Make those. And why don't we stay together as a big group for you guys to draw your amino acids in case you have trouble with that, because that's where you're going to run into issues. And then I'll put you into groups and like, then you can share your, what do you call it? Jamboard with each other. Do you guys know how to get to Jamboard? You go here. In Google, you just go to this. You find Jamboard in the Google apps. It's down below. Also, I gave you a link to Jamboard. Just use my link. That's your template. Just use that. Yeah. Just do that one. So you don't have to even find it. All right, everybody draw an amino acid now in Sketchpad. Do it. I draw 2 of them. Make one and then save it, and then do a flip one and save that. One with the R up, one with the R down. If you're having trouble, say something. I'll draw a few more just to show you that I'm doing it too. I'll do one with a mouse. Let's see how bad it is. I would like a bigger temp background, but oh well, that's what I got now. Okay. It's not so bad with the mouse. If you do it slowly, it works out nicely. Okay, then I'm going to crop it. Nice and tight. Maybe that's a little too tight. Okay. Go. Then I'm going to save it. Okay, by now you ought to be done, at least with one of them. I think I gave you a link to Sketchpad. I hope I did. Did I? Ah, Sketchpad right there, bam. Or you could just type Sketchpad. Know what I mean? Know what I'm saying? What's I doing here? This little light of mine, let it shine. All right, so you're going to need a couple of them with the R up and a couple of them with the R down. All right, that's enough time for drawing. Anybody have issues drawing and saving and finding that on Google Drive? Okay, I'm going to put you in groups now. I'd like you to, in your group, assign one person to be sort of like the boss. Not that you get to tell people what to do, but you're in control of the document of the jam. You share your Jamboard, you make a copy of mine, you share it with all the people in the group, and then everybody imports their amino acid. So there's going to be like a whole bunch of amino acids, like 8 of them. And then you align them up, scale them, make them smaller, all about the same size, and then line them up into a beta sheet. And then you use the jam pen to draw in the hydrogen bonds in like a pretty color. Good to go? It says my hard drive is full and it won't download. Yeah, it shouldn't have to go to your hard drive. It should be going to.
That's what mine said too.
It should be going to your Google Drive. So the jam won't, Sketchpad won't let you export. When I do this on my Chromebook, it goes right to my Google Drive. Is anybody successful with that, or say it louder?
Mine keeps popping up an error message and I don't know what to do.
Oh, has anyone been successful, or maybe the student Chromebooks can't do this? Because I can only test it on my teacher Chromebook. So in Sketchpad, you don't have the ability to download. You don't have a disk here.
Mine is working, but I don't know how it's working. So.
Oh, bummer. I'm wondering if we can use Cami for this. Trying to think of how to do this. All right, let's try this. I can copy this. I'm just going to copy that and see if I can paste it into like a Google Doc.
Can't you take a screenshot and copy it too?
Yeah, but I don't know how to crop it. Can we crop in Google Docs?
You can crop it when you're screenshotting
Oh,
it.
this is what it copied.
Yes.
So that didn't work. All right, so screenshot and then copy it in Google Docs. And then how do we save it as an image so we can import it into Jamboard is what I'm worried about. This was going to be nice. Okay.
And then
we're going to cheat. I have the solution just in case this happened. If you look at my instructions, where are my friggin' instructions? If you go down here, no, not here. If you go to Google Classroom, it says link to the folder containing the molecules. I have pictures already. I wasn't going to show you that yet because I wanted you to draw them, but apparently we're going to have issues downloading. So that's something we're going to have to still work out. And just go here. And look at that. There's an amino acid. You can just use that. Make a copy of that for you in your Google Drive. Put it in your AP Bio folder. Go to the other one, the normal one, and do the flip one and the normal one. Make a copy of it, save it. And then you can import those into Jamboard. So you're just going to use mine that look beautiful instead of doing your own. Sim did it a different way. We'll figure out how to do this so eventually you can get good at this stuff. But I guess today wasn't meant to be the day. Sorry about that. All right. How much time we have now? 13 minutes. I'm going to put you in groups, and then you create your beta sheet, hopefully before the end of the hour. And I'll kind of make big groups so I don't have to visit too many different groups. I'll do 5 groups of 5 to 6. So you can each import some and, no, let's do 6 groups. 4 to 5, yeah, let's do that. All right, good luck. Ellie, Amra, Anna, are you guys still here? You still show up on my screen. I don't know what's going on.
Sorry.
Are you able to get into the room or no?
I think so. Here.
Amra, are you still there? Amra. Amara, are you there? You have internet issues. All right.
Literally half an hour ago, I was like, this can't go.
Hopefully they'll go away by tomorrow, though, for in time for whatever, the basketball stuff.
Amara is stuck in the Ethernet someplace. She's supposed to be in this group, so she might still pop in. We'll see.
Okay, I'm sending it to both of you, okay?
Okay.
How's that beta sheet come along?
I just shared it with them right now.
Awesome. Thank you, guys.
Hey.
You guys able to share it with each other? You share the jam with each other? Yes.
I think Katie's sharing it right now.
See if you can whip up a beta sheet before the end of class. That'd be awesome. And one of y'all needs to mute. All right. See ya.
Hey, Mr. Volk.
Hello. Did the sharing work?
Yes. Yeah.
All right. See if you can whip up a beta sheet before the end of class.
Okay.
Awesome. And share it with me so I can see it.
I did.
Thank you. Did the sharing work for everybody? Sharing the Jamboard?
Yeah.
Yes.
All right, try to whip up a beta sheet before the end of class. See if you can do that. And then share with me, please. Share the Jamboard. Thank you. Were you guys able to share with each other?
Yeah, I shared mine with them.
All right, try your best to whip up a beta sheet before the class ends. Good luck. Could you also share it with me?
I did.
Thank you. Hello?
Hey, Mr. Volk.
Hello.
Were you successful sharing that jam with everybody?
Yes.
Yes.
We don't have a ton of time, but are you able to whip up a beta sheet before the bell rings? Well, bell.
Yeah.
All right. Also, if you haven't already, can you share it with me, please?
I did.
Thank you. Awesome.
Okay.
All right, from what I could see, those beta sheets were looking pretty good. Just make sure you end up drawing in the hydrogen bonds. Like you can circle the two atoms, or just draw like a dotted line between them. Get that going. Okie doke. All right, next week we'll try this again. We'll just do a real quick cell membrane building thing. And eventually we'll do the same thing with DNA, and hopefully it'll go a little faster. if we can't work out that Sketchpad saving issue, then we'll just do like pre-made ones, which is a little bit too easy, but whatever. Anyway, all right. Chapter 5 reading guide is due on Tuesday. So make sure you have that done. You're going to be answering questions on lipids that I haven't covered yet. And although I covered most stuff on lipids, almost done, I think. And you're going to be answering stuff on DNA that I haven't covered yet. So you'll have to read the book for that and look stuff up on the internet. But make sure that gets done. I noticed a bunch of you guys have figured out ways to type into PDFs. So I don't know what platform you're using for that. I know like Lumen PDF that comes with Google Drive can do that. Or you could use Cami or whatever. But I really like the people that did that, or I like the way you guys did that, where you're typing the answer right into the document. That was actually nice because it made it easy for me to read and everything. If you're handwriting it and then taking pictures, that's also fine. Ideally, if you could make me one file that you could turn in instead of like 7 or 8 separate pictures. If you could figure out a way to do that, that'd be great. Otherwise, it's okay to have just the separate pictures too. Anything else? What else we got to talk about? Anything else I'm forgetting?
No.
No.
It's Friday.
We must have a weekend. Not a happy, but have.
Have a weekend, yeah. Is Friday still a good thing, or is everything just a blur now? Fridays used to be a good thing.
Fridays are still good. We don't have school tomorrow.
You can sleep in.
I like Wednesdays now and Tuesdays.
Yeah. Although when we start school again, it sounds like we might start school again in less than a month. So then every day we'll be getting up early. So, all right, people, it's 1 o'clock. I think I'm supposed to end at 1. I don't know. Yep, 1 o'clock. See you guys next week. Bye.
Hey, Volk.
Yes.
What do we do today?
I did a review of beta pleated sheets, which is also on my videos that I posted. So you can watch my videos if you haven't already and get all that information. I posted a link to Max. You have a question too or no?
Yeah.
Okay. All right. I'll try to be fast here. Let me show you what I'm talking about here. In Classroom, I posted a link with all my videos here. So you can use this to find all the videos that you know, for stuff you might have missed. And then I started talking about lipids. So you would want to click on this one here. That's the video that goes over lipids. And then I think you were here for this part. That's the second part of lipids. You might have been back by then. I don't know when you came back.
Like 12, 30, 40, so not much.
Oh, got it. So that was just with that activity then. So you might want to watch both of these here and get caught up on lipid notes.
Okay.
So that's what you missed. So anything that I do in class, at least up to this point, I have videos pre-made that are already up here that are very similar to what I do in class that you can use. Are you okay?
I don't know.
You don't know?
Like, I got x-rayed, blood taken, a bunch of other stuff.
Well, what happened? Did you?
I've just been like nauseous and not eating, and then I threw up blood yesterday, so you know.
Oh, got it. Yeah.
Yeah.
Glad you went to the doctor.
Yeah, it's kind of a, you know, it's one of those things where it's like, I should probably go in.
Yeah. Once you throw up blood, that's a good sign to go in. You don't want to be stubborn anymore then. Well, good luck with that. I hope that works out.
Thanks.
All right, man. Hope you have a good weekend.
You too.
All right, Max.
Okay, so this doesn't have nothing to do with bio. Do you know anyone or do you want any guppies?
Do I want guppies?
Yes.
Yeah.
Okay.
Yeah, I can use them. There's actually 2 teachers here that would like them, Mr. Robertson also. You have a lot?
Yeah, I keep having baby guppies.
Yeah, they do that.
Yeah, I got 6 and I'll have like 40.
Yeah, nope. I would love them, but I don't know. I don't know how we would do that with. I'd have to meet you like outside of school or something.
Yeah, I can always give you to them. Give you when we go back to school if we ever going back.
If you don't mind waiting that long, that'd be probably the
Yeah,
easiest.
that's
That might
fine.
be the easiest. You just bring them in the morning then.
Yeah.
So yeah, that'd be cool.
Okay.
Take some pictures of your fish tank with the guppies and then send them to me. I'd like to see them.
Okay, I will.
All right, cool. Have a good weekend, man.
You too. Bye.